Hi, everyone, and welcome back to the channel. So in this video, I'm going to talk to you about population genetics. So population genetics is the study of changes in phenotype and allele frequency within a population. So hopefully if you're watching this video, you're familiar with the concepts of genotype and phenotype. Genotype is what the genetics underlying a particular trait are. The phenotype is the outward appearance of that particular trait. So in general, if we're looking at the rate at which a given phenotype shows up in the population, that can also give us an indication of what the allele frequency is that's controlling those particular phenotypes. And in terms of population genetics, allele frequency is how we're going to measure evolution. We're going to determine how much evolution is occurring, for example. So for example, in a study done in the country of Jordan, they were looking at the allele frequencies for the three alleles that control the ABO blood grouping. They found that, for example, the frequency of the A allele in that population was 26.1%, the frequency of the B allele was 13.4%, and then the frequency of the O allele was 60.5%. So if they go back and they redo this study in 10 years, 20 years, however it is, and we see that those frequencies have changed significantly, that would be an argument to say that, yeah, evolution is occurring with respect to that particular group of alleles within that population. And this shouldn't be something that's surprising. Whenever we look at a population of any species, we're going to see variation, right? So it's the study of the changes in how those variations, what level those variations make up the population that we're talking about. So understand that when we're talking about this from a genetic perspective, we are talking about heritability. Heritability refers to the fraction of phenotypic variation that can be attributed to genetic variation. Obviously, there are some changes in phenotype that are not controlled at the genetic level. People working out, people going to get tan, animals that have other sort of physical changes that happen to them throughout their lifetime. But we're specifically focusing on those traits that are heritable because those are the only ones that natural selection, as well as other evolutionary forces such as genetic drift, bottlenecking events, that those things can work on. They can only work on heritable traits. Now, one thing we do have to acknowledge is that a real allele frequency can change for many reasons. This is kind of what I was alluding to. So yes, we can get changes in allele frequency that are directly attributed to natural selection. We'll talk about those in a minute, but what are some other reasons for why allele frequencies may change within a population? A lot of these can be tied to a phenomenon called genetic drift. And genetic drift, it refers to the change in allele frequency that happens through chance. So give you just a quick example of what genetic drift might look like, how it might occur in a population. Let's look at this population of rabbits. We can say that there's only 10 rabbits in this population, or we can say that, for example, each of these rabbits represents 10 rabbits. But either way, we look at this population here, and we can see that we can calculate the allele frequency to be about 0.5 for the B gene and 0.5 for the Q allele of that, or the little b version of that allele, right? So we've got big B dominant, little b recessive. Either way, if we add these up, there are 50% of the B's you're going to count here are going to be big B's, 50% of the B's you're going to count here are going to be little b's. And as a result, we can say that the gene frequency of big B is 0.5, the gene frequency of little b is 0.5, okay? Now, notice that phenotypically, this doesn't perfectly line up, right? Why? Well, because this is a situation of complete dominance, where any version of the big B is going to give you the dominant allele, and you need to be homozygous recessive for the little b allele in order to have the albino phenotype. But regardless, we're going to say that it's just this proportion of the population that ends up reproducing in this generation. Okay, so we've got a couple of heterozygous individuals, a couple of homozygous dominant individuals, a few heterozygous individuals. These are the only ones that make up the breeding population for this particular generation. So when we look at the second generation of these rabbits that result from this particular mating, or these particular matings, we see that now the frequency of these alleles has changed. And as a result, the big B allele assigned the variable P here, you'll see why in a little bit, now is at 70%. It's at 0.7. So now if we add up all the B's, 0.7 of those, 70% of those are going to be big B's. And now we have only 30% of the population or 30% of the allele frequency being little b's at this point. Okay. So now we can see there has been change. But because we're just saying that this change has to do with who happened to reproduce last time, it wasn't dependent on, hawks eating more of the albino rabbits, or, the albino rabbits are not able to acquire food for some reason. We're not saying it has anything to do with natural selection. Just out of sheer random luck, these are the 5 rabbits that reproduced. These are the offspring that they have. And now all of a sudden, the next generation, for whatever reason, it's not tied to any type of natural selection. Now it's just these 2 individuals who reproduce. Therefore, the entire 3rd generation is all big Bs. They're all homozygous dominant. Now, the frequency of the big B allele is 100. It's 1. It's 100%. The little allele, the little b, has now completely gone away from this particular population. Okay? That can happen strictly through genetic drift, just through sheer randomness. And if that happens, we do get changes in allele frequency that are not tied to adaptation. They're not tied to natural selection. Okay? One of the things we have to note is when we talk about genetic drift, smaller populations are significantly more susceptible to genetic drift than larger ones for this very reason. If we're talking about a population of just 10 rabbits, and 50% of that population happens to be the ones that reproduce, well, there's a better chance in a population of 10 than there would be in a better population of 100 that it's only these individuals who reproduce. And slowly we see actually just 3 generations, fairly quickly, we see the disappearance of the little b allele. If this were a population of 100 rabbits or 1,000 rabbits or 1,000,000 rabbits, now it becomes much less likely that this little b allele disappears over time. But this is just an extreme example of that. So understanding when we talk about genetic drift, we're talking about changes in allele frequency that are happening just to random chance. They're happening just to luck, essentially, and not tied to any sort of selection pressure. Some other reasons where we may see a change in the population also are bottlenecking events. So I have talked about bottlenecking events in other videos of mine, but I'll kind of rehash that here. So bottlenecking events occur when a significant force acts on a population to drastically reduce the size of that population. Okay. Many times in nature, bottlenecking events are natural disasters. These could be on the grand scale when we talk about mass extinction events that have happened in evolutionary history, or they could be something as significant or something as less significant as like a drought, a tornado, a flood, something that impacts a population where it's not something that natural selection has a chance to really act on. It's a massive event that isn't particularly selective and just wipes out a huge percent of the population. In doing so, you reduce that population size significantly. And maybe where you had 1,000 rabbits, maybe you're just down to these 10. Maybe there's a 3rd allele that used to be in this population that no longer exists in the first place. And now we're down to these 10 rabbits. And these 10 rabbits now make up the entire breeding population for this group of rabbits. And as a result, we can see that allele frequencies can fluctuate wildly in this population. So bottlenecking events can be natural disasters. They can be mass extinction events. The other time we see bottlenecking events is when we get like accidental migrations. So I've talked about in other videos how island biogeography is really interesting, right? Because quite often it's only a handful of birds that make it to that remote island, that remote oceanic island that start that population, or a handful of, turtles that were able to raft, or tortoises that were able to raft across a wide channel. These can also serve as bottlenecking events. So in general, a bottlenecking event is any type of event where a population goes from a relatively large size to a relatively very small size. And in doing so, those allele frequencies change drastically in that process. So again, we could be looking at an example here where these 10 rabbits are from a much larger population of rabbits, but for some reason, they've now become geographically isolated. Maybe they're on an island on a lake that's recently swollen up, right? That lake is now swollen and they used to have a connection to the mainland. Now they're just stuck on this island because, there's a massive flood, or we built a dam and now what used to be, a low-lying area with a hill in the middle, now the hill is the only thing you can see and the rest of it is a lake, a man-made lake, because we built a dam to dam up that river. There's lots of ways this can happen. But these bottlenecking events, by reducing the population, can drastically impact the allele frequency within that population, and by reducing the size, they make it much more likely that genetic drift is going to have a significant impact on allele frequencies over time. Some other examples could be gene flow. So gene flow is another way in which we get introduction of new alleles or the exiting of existing alleles from a population. So gene flow is another way in which allele frequencies can change without adaptive selection occurring, without natural selection, okay? So gene flow occurs when animals enter and leave a population through migration. We can say the migration of gametes or individuals. So for example, with what I'm showing here, we have a mountain range. There's a small pass in which deer infrequently cross. So there's a western deer population, an eastern deer population. You can see in this example, they've given the appearance of different coat colors to indicate their groups. You can see while there are some darker colored individuals here, the majority of them are light colored. The situation is the exact opposite here, but let's say every year during the summer, there's a few months where this pass, or maybe even a few weeks, in which this mountain pass is open, and some deer invariably cross back and forth during that time. And in doing so, they bring themselves as well as their gametes and alleles into this population, and this gene flow can disrupt sort of the balance of allele frequencies by introducing new alleles or by sending some current alleles to a different population. Plants, on the other hand, many plants are called broadcast pollinators. For example, trees or flowers that use wind. So they just send their pollen everywhere. And in doing so, they can send pollen or their gametes outside of their geographic range, and they can now enter into distant populations. And in that sense, they can introduce new alleles to their population, or if those alleles have left the breeding range, the normal breeding range for that particular flower, those alleles have now left the population and are now belonging to a new population. So this can lead to changes in existing allele frequencies or the appearance of entirely new alleles through this mechanism. And then we also have to understand that mutations always happen, right? They happen randomly. And when a new mutation shows up in the population, that can alter the allele frequency, assuming that the individual with that particular mutation is able to find a suitable mate and produce viable offspring. Another thing outside of natural selection is called non-random mating. So non-random mating can occur for many reasons. First off, sexual selection. Now we'll come back to sexual selection in a minute. Sexual selection is actually a form of natural selection, but recognize the fact that we have to assume in many populations that the mating selections are not random. There's also something called assortative mating. So this occurs when individuals choose mates based on their phenotypic similarity. So maybe you have, two, maybe more phenotypes with respect to a particular trait in your population. But for this particular species, those with matching coat colors tend to reproduce with each other. So if you have, we go back to the deer example, while there are some dark colored individuals in this lightly colored population, perhaps these two are going to mate with each other at a higher frequency simply because they look more alike than they're less likely to mate into the lighter population. So this would be an example of assortative mating. It's non-random. They're selecting for something. Or it could be physical location, right? If you have a population that's spread over a very, very large area. So for example, if we look at fish species that reside in the Mississippi, there are no geographic barriers to those fish being able to reproduce with each other. But if one fish lives down in New Orleans and another one lives up in St. Louis or way up in, Minnesota, it's very, very unlikely that those two individuals will find each other and mate. They're likely to mate with individuals in the population that are geographically closer to them. You might also find that, in fact, there are geographic barriers that also make it so that while members of the same species may be close in terms of their physical proximity, there are geographic barriers that prevent them from interacting. And as a result, the mating isn't as random as we'd like it to be, simply because they're going to mate within the population they can get to and not necessarily other members of the same population that are harder to reach due to their physical location. Also, we have to reckon with the fact that environmental variance matters. So abiotic factors can often influence the phenotypic variance within that particular population, i.e. allele frequency. A great example of this would be clines. So For example, we talked about those fish that are in the Mississippi River. The fish that are down in the southern portion of the Mississippi vary significantly in terms of their phenotypic appearance for those that from those that are significantly further north in the Mississippi because the quality of the water changes, right? So we do see that there are changes to allele frequency in those populations, and environmental variance might contribute to changes in allele frequency within that population depending on where you're looking. We also know that some species, particularly in the reptile group, tend to be exhibiting temperature-dependent sex determination, which is really wild when you think about it, but the temperature at which the egg is incubated often changes whether the individual will be a male or a female of the species, and as a result, the abiotic factors do contribute to the appearance, at least of those particular alleles. Are they the male allele or the female allele to make that individual male or female, which is also very interesting, or at least it changes the phenotype in some species to being male or female, depending on the temperature at which they were incubated while they were still an egg. So there are lots of reasons outside of natural selection why we may see changes in allele frequency within a population. But what if natural selection is the culprit, as is so often the case? Well, we call this adaptive evolution, right? Because this is evolution in response to natural selection. And as you hopefully know from some of my other videos, natural selection is the only mechanism by which adaptation can occur. So adaptive evolution occurs when natural selection is acting on a population. And in doing so, selection pressures that act on that population are going to favor those with higher reproductive fitness, also known as evolutionary fitness. So when we talk about reproductive or evolutionary fitness with respect to individuals, we're talking about the ability of that individual to survive and reproduce. Now, we need to dispel the commonly held myth that natural selection is about survival of the fittest, and then using the connotation of fitness to think about fitness in more of a human construct, which is bigger, faster, stronger. Reproductive fitness does not always mean bigger, faster, stronger. In fact, in some cases, it could mean the exact opposite, depending on what the environment dictates. And that's one of the things that we have to understand about reproductive fitness is that reproductive fitness is relative. It can change over time. So reproductive fitness is a function both of an individual's genetic makeup and the environmental makeup. And while individuals can't change their genetic makeup, the environment can change. And what that means is sometimes what's good today in terms of reproductive fitness may not be good tomorrow. And things that aren't necessarily favored today in terms of reproductive fitness mean that they could be favored later on if certain conditions change. All right. I have another video on this, so I'm not going to belabor this point, but in terms of the types of selection we observe, there are three major classes. There is stabilizing selection. In stabilizing selection, natural selection favors it favors sort of a middle phenotype, a middle ground, neither of the phenotypic extremes. So, for example, here, there is sort of an ideal clutch size for various birds. If you have too many eggs, some of those chicks are going to starve because you can't feed them. If you have too few eggs, on the other hand, you run the risk of no viable offspring occurring. Smaller clutches, you're simply going to have some amount of attrition. Some eggs just aren't going to pan out. Well, if you only lay one or two eggs and one or two of those eggs don't hatch, you're not going to have any offspring. If you have too many, you might not be able to feed them. So there's an ideal number somewhere in the middle. Directional selection, where one phenotypic extreme is favored over the others. You've probably heard of the peppered moth situation before. The original population had this kind of phenotypic appearance where they were white with black flecks. As coal began being burned in the Industrial Revolution in England, all of the trees became soot covered from that coal. And all of a sudden, it became much more favorable to be of the black variant where you can blend in on those trees. And then now that England's cleaned up its act and got away from coal burning power plants and coal burning for heat in homes, it's now shifted back the other way. We're playing between the phenotypic extremes. And then we have diversifying selection. And in diversifying selection, both phenotypic extremes are favored over the original. All right. So, for example, here in this hypothetical population, gray rabbits are better able to blend in or what we would call the Himalayan phenotype. They're better able to blend in with rocky environments than either of the middle, which is albino. White rabbits stick out very heavily, whereas either one of these is a better situation. I have a video on modes of selection. I would go back and watch that if you want more of an explanation. But what I want you to understand is that when we're talking about adaptive evolution, we're talking about a response to selection pressures. This is the natural selection that you're accustomed to here. This also includes sexual selection and what we call frequency-dependent selection. So sexual selection is a mode of natural selection where in certain species we see sexual dimorphisms. These are traits or characteristics that differ between the different sexes. So in the case of peacocks and peahens are very choosy for peacocks that have giant fantails with multiple eyes on them. So it behooves males that they want to reproduce to have these giant displays. So sexual selection has naturally selected for males that have this particular, these particularly bright displays, and females are choosy for those. There are lots of examples of this. Go look up bowerbirds. Again, I have a video on this type of selection, on sexual selection. Go watch it if you need more about that. There's also something called frequency-dependent selection, which is very interesting. These are what are called side-blotched lizards, but they can have 3 different types of necks, orange-colored, blue-colored, and yellow-colored, and their reproduction strategies are actually very very interesting. So orange males are much more aggressive than the other 2 colors, and they will mate with females, but try to mate with as many females as they possibly can. On the other hand, blue-throated males, they are less aggressive, but when they mate with their partners, they tend to mate for life, and they tend to pair bond much more powerfully. And then you have the yellow-necked ones. The yellow-throated ones look a lot more like the females of the population, and as a result, they can have side copulations with females that are in committed relationships. And sneak these side copulations in. So the reproductive strategies are very different. What's interesting is the population sort of changes. Like there's a giant genetic rock, paper, scissors event happening. So, for example, in a population dominated by blue males, orange males have an advantage. Orange beats blue because they will pair with females that the blue-throated ones would like to, but then they'll also go off and pair with other females. So males get outcompeted there. On the other hand, males outcompete the yellow-throated ones because the yellow-throated ones are looking for side copulations. They're essentially looking for females that are willing to be polyamorous, whereas the males are more committed in that relationship. All right, so they can hopefully pair stronger with those females and prevent these side copulations. Interestingly, the yellow-throated ones are actually able to outcompete the orange ones because while these guys are reproducing with lots of different females, they're not necessarily in a relationship, like a committed relationship to those females. So those females don't feel as bad going off and side copulating with the yellow-throated ones. So you get this sort of fluctuation in the population where when blue dominates, orange will start to take over. As orange takes over, the yellow will start to take over against the orange, and then the blue steps in and outcompetes the yellow. So you get this sort of revolving door in this population based on their ability to reproduce. This is what we call frequency-dependent selection. I don't talk about this in other videos, so I wanted to take an opportunity to talk about this here. But let's circle all back to what we're talking about in general, right? We're talking about population genetics. So what I've talked about in the first part of this video is reasons why allele frequencies change. These are all reasons. It could be genetic drift. It could be some of these other, genetic drift phenotypes, or it could just be natural selection acting on a population. But how can we actually tell if a population is in fact evolving? Or another question would be, can we determine the allele frequency within a population based on phenotype? And in the early part of the 20th century, shortly after the modern synthesis between Mendelian genetics and Darwinian evolution got fused together, the modern synthesis, two scientists became interested in this. You have an English mathematician named Gottfried Hardy and a German physician called Wilhelm Weinberg, who developed the concept of equilibrium to describe the genetic composition of a given population. All right. This leads to something called Hardy-Weinberg equilibrium. And what they basically state is that the allele frequency of a population is stable unless some evolutionary force is acting upon it. Okay? It's a little bit of Newton, right? Newton comes out and says, describes inertia, right? An outside force remains stable. I'm sorry, an object remains stable or doing whatever it's doing unless acted upon by an outside force. They apply this concept to population genetics and saying all things being equal, unless evolution is happening in some way, we should expect the allele frequencies within a given population to remain constant. Now, just like Newton said, like in a vacuum, right, there's these caveats, these theoretical conditions we have to be able to meet in order for Hardy-Weinberg equilibrium to actually work. And here are their conditions. First, the population needs to be of infinite size. So what they're saying here is you need a really big population, right? We don't often find vacuums in nature, except maybe in space, right? So Newton had to say in a vacuum. This is kind of their in a vacuum caveat. The population has to be huge. It has to be of infinite size. We also have to assume that there are no migrations, no emigrations, so nobody's entering or leaving the population. Gene flow's got to be gone. No mutations, which we know isn't going to happen, and there cannot be selection pressure applied to a given genotype, right? There's got to be no sort of selection pressure to get adaptive evolution to happen, right? So we know, and also all matings have to be random. So what we have to understand is this theoretical set of conditions likely to be met in any natural population? And the answer is probably no. All right. It probably can't be met by any real world population, but it does serve as a very useful model to compare populations against. Like, this is the ideal. If we see a deviation from this, we can assume that some amount of evolutionary pressure is being applied in some way. Now, one thing we have to stipulate, large populations, humans, for example, do actually approximate this state. They actually do. Across many of our alleles, we can see the results are essentially in line with Hardy-Weinberg equilibrium. But what this, and the big thing here is this allows us to assess populations over time and say, okay, are they changing? And with respect to which alleles are they changing? Because there could be some alleles in a population that aren't, while others are. Okay, so here's how this works. All right, they basically say we're going to assign the dominant allele in any population as P and the recessive allele as Q. And we're going to talk in terms of strict dominance just to keep this as easy as possible, because we're just trying to understand the concept of Hardy-Weinberg equilibrium. Okay, to calculate the value of P and to calculate the value of Q, all you have to do is count the number of alleles in the population and divide by the total number of alleles counted. Homozygous individuals count double because remember, they have 2 copies of the allele. So let's look at this. We look at a population, and let's just say we have a way of determining this, whether it's phenotypically, whether we have the ability to assess genotypes. Again, a hypothetical example. We are going to look at 500 individuals within this particular population, and we're going to look with respect to a gene that has 2 alleles, big Y and little y. Why? Where big Y is the dominant allele and little y is the recessive allele. It looks like in this particular example, we're going to use P's and we're going to use P color. Big Y gives you yellow, little y gives you green. The heterozygote, because this is complete dominance, is yellow despite only having 1 big Y allele. We calculate from this that we have a genotypic frequency we have 245 individuals that are homozygous dominant. We have 210 individuals who are heterozygous, and we have 45 individuals who are homozygous recessive. All right, little y, little y. We now add all those together. We say, what is the number of alleles in the gene pool? So first we have to take the 245 here for the homozygous dominant, and we have to double it. Why? Because there's 2 big Ys in there, which means there are 490 big Ys in this group of the population. And we have 210 individuals that are heterozygous, so there are 210 big Ys there for a total of 700. We do the exact same thing. Again, 210 individuals are heterozygotes, which means there are 210 little Ys as part of that population. And then we have 45 individuals who are homozygous dominant or homozygous recessive, and that means there are 45 times 2, 90 little y alleles here for a total of 300. We now divide those totals by the total allelic frequency 700 divided by 1,000, the allelic frequency is 0.7 for P. 300 divided by 1,000 is 0.3. The genotypic frequency for the little y allele, or Q, is 0.3. All right, so allele frequencies of 0.7 for P, 0.3 for Q. Okay, so we can then now apply that to the Hardy-Weinberg formula, which says that, okay, well, basically we're just sort of calculating the frequency of the Punnett square. So what's the frequency of being homozygous dominant? 0.7 times 0.7 gives you 0.49. What's the frequency of being a heterozygote? Well, it's 0.3. It's 0.3 times 0.7, but you have to double it. Why? Because there are two ways you can become heterozygotes. And then again, we just square this to get 0.9, and you can see that the predicted frequency for any offspring in this population would be 0.49, 0.42, and 0.09. Again, we're just taking the numbers we saw and plugging it into the equation. Okay, that would be the predicted frequency we would expect to see from the offspring. All right, here's the starting population. Here's what we're calculating through our Punnett squares and our product and sum rules. And this is telling you what the allele frequency should be in the offspring if there is consistency, right? If we go from one generation to the next generation and we are in Hardy-Weinberg equilibrium, we should get the exact same proportions back out, right? Because Hardy-Weinberg equilibrium says that if there is no outside influence, we should get the same allele frequencies back out generation after generation after generation. Now, if we come back and look at this subsequent generation, we find that the allele frequencies are not matching the predicted allele frequencies. Then there must be some type of evolutionary pressure being applied to this particular population. And as a result of that evolutionary pressure, we are seeing changes in allele frequency. Now, note, that doesn't specify that it is a selection pressure. It doesn't specify that it's genetic drift. It could be any of those scenarios that we talked about. It just simply says that this species is changing. Okay, then you have to go back in and find out why, right? You'd have to go back in and take a look. But if you see the same allele frequencies generation after generation after generation, it's pretty safe to say that with respect to those particular alleles, this population is in Hardy-Weinberg equilibrium. But we can also use the Hardy-Weinberg formula to predict allele frequencies. So let's end by giving you an example of how we can use this to predict, because this is more likely what you might be doing. Okay, so let's say that we discover that there's a particular disease or a particular condition in the human population. We just discovered this, right? And we find out that it's linked to this particular allele. People who are homozygous for this particular allele, will say it's a recessive allele, are going to be affected by this disease. But now the question becomes like, wow, well, we know that 0.04% of the population is affected by this autosomal recessive disease. The next question becomes that, man, what percentage of the population then are carriers for this particular disease, right? Like, how many people do we have to like really alert? Like, hey, we've recently discovered that this particular condition, it's very rare, but it does exist. And here it's a genetically inherited condition. It's not something spontaneous. It's not something environmental. It's not from exposure to a virus or something like that. It's genetic. And now we got to figure out what percent of the population maybe doesn't have the disease, but's carrying those disease-causing alleles. Well, we can do this using the Hardy-Weinberg formula. So how does this work? So first off, we do know the phenotypic frequency of this disease, right? So we can calculate Q, because we know in order to have this particular disease, you have to have this particular genotype, little e, little d. All right, so we're going to calculate Q this way. So how do we do this? Well, we know then that Q squared is 0.04% or 0.0004. That's the fraction of the population, right? Hopefully we're understanding that percentages per 100. So to make this what the frequency is, we just have to divide by 100 to get that number, 0.004. So which means Q squared is 0.0004. Which means Q is the square root of that number, which is now 0.02. Okay, so Q is 0.02. Now that we know that, we can determine P, because we know that based on Hardy-Weinberg, we know that that P plus Q has to equal 1, right? Because they're fractions of a whole. So we know then that 1 minus Q, right? If P plus Q equals 1, then 1 minus Q must equal 0.98. So we know that the frequency of P is 0.98. So then how can we calculate the number of carriers? Well, it's simple. We know that the heterozygotes are equivalent to 2pq, right? Because p squared, because if we go back to our original Hardy-Weinberg formula, we go back here, we know that p squared plus 2pq plus q squared must equal 1. Well, we know that p squared is 0.98 squared, which is 0.96. We know that q squared over here is 0.02. So we do the equation. Q is 0.02. So 2 times PQ, 2 times 0.98 times 0.02 equals 0.04. So we know if we multiply 100 that 4% of the population is a carrier of this allele. All right. So 0.04% of the population, a very, very tiny fraction, is actually affected. But more broadly, we now know that about 4% of the population, which isn't that small, are actually carriers. So people who have an individual in their family that has this disease might need to go check, is there probably, or they're very likely part of this 4% of the population that's carrying around this allele. So what I want you to understand is that Hardy-Weinberg equilibrium is sort of this hypothetical situation that relies on a bunch of conditions that most likely cannot be met, but can be closely approximated in large populations, large natural populations. And if we assume Hardy-Weinberg equilibrium is existing in these large populations, we can then assess using the Hardy-Weinberg formula and our understanding of mathematical concepts that we can begin to understand by saying, okay, here are the observed allele frequencies right now. Do they match what Hardy-Weinberg equilibrium says they should match? And if they don't, that tells us that there are some evolutionary force acting on that population. If they do match the theoretical calculation from Hardy-Weinberg equilibrium, then we can say then, okay, this population is no longer in Hardy-Weinberg equilibrium because it has deviated from those predicted values. This population is evolving. That then leads to a series of investigative questions that you can ask. What is the evolutionary force? What is acting? Is it genetic drift? Is there something going on with the mating within these species? Is it sexual selection? Is it another form of natural selection? Is there some selection pressure acting on this species to cause them to adapt? You can begin asking those questions. Or if you're looking at something from sort of the reverse side of like, okay, we've discovered this new phenotype, or we now understand that this phenotype is the result of heritable traits. Okay, well, can we figure out what percentage of the population that we can expect to be carriers of this particular allele? Or if we know one, we know the amount of the dominant allele, do we know the amount of this recessive allele that's running around our population? We can calculate all these using the Hardy-Weinberg formula to have some sort of predictive power. Okay. So while Hardy-Weinberg equilibrium is sort of this theoretical situation that we can't meet all the characteristics for in nature, it gives us something to compare to so we can begin to understand how much evolution is happening within a population. So I hope this video helped you understand population genetics, what we're looking at in terms of population genetics, and then how we can use the Hardy-Weinberg formula and the Hardy-Weinberg equilibrium to help understand what's happening within a species with respect to a given allele or multiple alleles if that's what you're interested in. So I hope you found this video interesting. I hope you found it helpful. I hope it helps you understand how to apply the Hardy-Weinberg formula and understand what the Hardy-Weinberg equilibrium is supposed to be telling us about populations. And now we can use that to understand what's going on within populations. I hope you enjoyed the video. I hope to see you watching other ones of mine, and I will talk to you real soon. Bye.